Okay, so in the last class we are discussing about vaccines. Vaccines and their applications in animal infections. So we are discussing about what is a vaccine, what are the types of vaccine, and what are the vaccines which are available for animals, vaccines which are available for the animal diseases, right? Bhavyashree, நீங்க எடுக்கிறீங்களா? Okay. If you want, you can share the screen also.
Sir, screen is visible, sir?
Yes, visible, visible.
Okay. Good morning. Today I'm going to talk about vaccines and their applications in animal infection. Vaccine. What is vaccine? Vaccination is biological preparation and immunization of a domestic livestock or wild animals. They are used to protect pets from highly contagious and deadly diseases. Types of vaccines. Vaccines are dead or inactivated organisms or purified products derived from them. There are several types of vaccines currently in use. These represent different strategies used to try to reduce risk of illness while retaining the ability to induce a beneficial immune response. Now we are going to see the types. There are three types: killed, attenuated, toxic. First we will see about killed. Some vaccines contain killed but previously virulent microorganisms that have been destroyed with chemicals, heat, radioactivity, or antibiotics. They generally provide a relatively short period of immunity. Examples are the influenza vaccine, bubonic plague vaccine, polio vaccine, hepatitis A vaccine, and rabies vaccine. Attenuated. In attenuated vaccine, the immunizing agent is an organism such as virus, bacterium, or parasite, which has been developed to stimulate the production of the appropriate antibodies without causing the disease. Live vaccines are particularly effective in providing long-term protection because they are more powerful stimulants to the immune system. They are also more versatile in the route of administration. Toxic. These toxic vaccines are made from inactivated toxic compounds that causes illness rather than the microorganism. Example of toxic-based vaccine includes tetanus and diphtheria. Toxic vaccines are known for the efficacy. Not all toxic for microorganisms. For example, Proteolus atrox toxic is used to vaccinate dogs against rattlesnake bites. Phenylene vaccine. This phenylene vaccine is used for cats. In cats can be prevented, controlled, or alleviated through effective vaccination. These include phenylene calcivirus, phenylene leishmaniavirus, phenylene rabies. Canine vaccination. These vaccinations are scheduled for dogs, including core and non-core vaccines. The important canine diseases are canine Dastember virus, canine parvovirus, canine adenovirus, canine leptospira, canine parainfluenza virus. Rabbit vaccination. Vaccination of pet rabbits is an important part of proactive approach to preventive health care and is widely recommended to protect rabbits from two very serious viral infectious diseases, myxomatosis and rabbit hemorrhagic diseases. These diseases can result in rabbits needing prolonged intensive care treatment. And in many cases, the most serious viral disease of rabbits are fatal. Farm animal vaccination. Farm animals are represented by a variety of different species from cattle, sheep, pigs and poultry to farmed fish. This means there is a correspondingly diverse range of potential disease threats and vaccines available to protect farm animals. Conclusion: vaccination is an effective way to prevent or reduce diseases in pets, farm animals, and fish. Vaccines are available for a range of important bacterial, viral, fungal, and parasitic diseases. Although it is not always technically possible to develop an effective vaccine, the animal vaccine medicine sector continues to invest and innovate in vaccine technology to support endemic and exotic disease prevention and control. Vaccines are therefore part of a wider range of animal medicines available to prevent or treat diseases. So,
Yes, ma'am. Okay, thank you.
that's all, sir.
Okay, thank you. Okay, so that's all of vaccines. So if you have any doubts about vaccines,
No, sir.
clarify. Okay. Okay, so we have come to an end of unit 3, right? So we'll have some brief description about unit 3 and then we'll move on to unit 4. So unit 3 we have seen about therapy of animal diseases. So in that we have seen in the treatment of animal infections, recombinant cytokines and monoclonal antibodies in therapy, gene therapy, and then vaccines. Okay, so these are the things which we have seen in unit 3. So any doubts in unit 3? You wanted to go again means also we can see in the unit 3 again any topic and then we'll move on to the fourth unit. Doubt irukka? Joel? Sandhya?
Yes, sir.
Yes. Any doubt in your third unit?
No, sir.
Okay. So we'll move on to the fourth unit. So fourth unit is micro manipulation of embryos. So what is micro manipulation technology? Okay. So what is micro manipulation technology? Equipments used in micro manipulation, and then artificial insemination, in vitro fertilization, the different techniques of in vitro fertilization, micro manipulation technology we are going to see in this unit 4. Okay, so micro manipulation. So what is micro manipulation technology? Okay, so there are two methods in fertilization, as we all know. One is an artificial method and one is a natural method of fertilization. Okay, so in these two methods, which we are going to see is an artificial method where we are going to fertilize in vitro. Okay, so one of the technique is micro manipulation technology. Here, the procedure will be performed using an microscopic instruments to manipulate the eggs, sperm, and embryos. Okay, the main technique here in the micro manipulation technique involved here is we will be using a microscopic instruments to manipulate oocytes, sperm, and embryos. Okay, now they are using this kind of micro manipulation technology will be remotely operated, and it will be inoculated and inoculated and manipulated, okay, using then microscopic needles. Okay, for example, like eye surgery. In eye surgery, you may been seen யாராவது வீட்டில் இருக்கவங்களுக்கு இந்த கேட்ராக்ட் ஆப்ரேஷன் பண்றப்ப வந்து பாத்தீங்கன்னா they will be showing it in a live TV. Okay, so like that, like that, and like a remote instrument we will be using for this micro manipulation technology. Okay, so these microscopic techniques are designed to increase the chances of a successful in vitro techniques. That, for example, in vitro fertilization. You may be heard nowadays there are more hospitals are evolving for this in vitro fertilization techniques. கேள்விப்பட்டிருக்கீங்களா IVF? Akshaya, Bhavyashree, in vitro fertilization. கேள்விப்பட்டிருக்கீங்களா இல்லையா?
Yes, sir.
கேள்விப்பட்டிருக்கீங்க. Okay, so these are the microscopic techniques which are designed to increase the chances of a successful in vitro fertilization. Okay, so normally when they are going for the fertilization process, normally the animals in the cell culture ல சொல்லியிருக்கேன், cell culture னு போறப்பே வந்து பாத்தீங்கன்னா they will be doing in triplicates. Okay, triplicates or more than that also they will be doing to increase the chances of success rate. Okay, they will be taking the 3 triplicates, and in that they will be choosing one. Okay, so when we are going for this micro manipulation technology, the chances of success rate will be increased. Okay, and then so why these kind of micro manipulation technology we are using? Okay, during fertilization inside the human body, for example, normally every 1 second. Can you see the slide?
No, sir.
Can you see now?
Yes, sir.
Okay. Okay, so in micro manipulator, which is a device used for the micro manipulation technology. Okay, like our pipette, it will be in a micro size, so that will be attached to an instrument. Okay, so we will be doing this micro manipulation technique with the help of microscope. Okay, high-end microscope and inverted microscope they will be using. Okay, so you know about inverted microscope. What is inverted microscope? You know about inverted microscope? தெரியுமா தெரியாதா? கேள்விப்பட்டிருக்கீங்களா? ஸோ தலைகீழா இருக்கும். ஓகே, நார்மலா வந்து லைட் வந்து கீழே இருக்கும், மேல லென்ஸ் இருக்கும் இல்லையா? எங்க லென்ஸ் இருக்கும் நார்மல் மைக்ரோஸ்கோப்ல? லென்ஸ் மேல இருக்குமா கீழே இருக்குமா?
மேல சார்.
ஸோ அதே வந்து அப்சைட் டவுன்ல இருந்து they will be attached with the camera. நான் காமிக்கிறேன், நெக்ஸ்ட் இந்த வீடியோல காமிக்கிறேன். So it may typically consist of an input joystick. So அந்த joystickல தான் வந்து they will be operating the microscope. Okay, so all these things will be remotely operated, reducing the range of movement, and the movement of this the joystick movement இருக்கிறதுனால you can have a micro movement. ஏன்னா நம்ம கையில குடிச்சு பண்றப்ப it will be shaked up, and that micro movement you cannot do. Okay, ஏன்னா அந்த செல்லுக்குள்ளார பண்றப்ப it will be very sensitive. Okay, so for that we will be using this micro tools. Okay, and micrometers are usually used in conjunction with microscope. It is a method used in the isolation of a pure culture technique. Okay, the main thing which we are experiencing here is more number of success rate, so less contamination will be there. Okay, so this is a microscope they will be using. So micro manipulator have been built which permit one to pick out a single cell from the mixed culture. Okay, so ஒரு கல்ச்சர்ல இருக்க ஒரு பிளேட்ல இருக்கிற கல்ச்சர்ல இருந்து சிங்கிள் செல்ல இருந்து also you can single cell also you can take from a culture. Okay, so this instrument is used in conjunction with microscope to pick a single cell, particularly bacterial cell, from hanging drop preparation. Okay, so this is about a series of hanging group. Okay, next. So this is a micro manipulator instrument. So which will be attached with a microscope. So lens வந்து மேல இருக்கும், sorry கீழே lens இருக்கும், மேல வந்து it will be attached with a camera. Okay, so by seeing the visual they will be doing. Okay, so it will be attached with a TV or monitor, it will be attached. Okay, so this cell is drawn into the micropipette by gentle suction, and it is transferred to the large drop of the sterile medium. Okay, when the number of cells increases in that drop as a result of multiplication, so this yield a pure culture of a required microorganism. Okay, the example on the microorganism ku vechirken. Okay, so this is the instrument setup. Slide maarudilla?
Yes, sir.
Okay. Okay, so advantages of this method are, which can be reasonably sure that the cultures come from a single cell and one can obtain strains within the species. Disadvantages: equipment is expensive, its manipulation is very tedious, and it required a skilled operator. Okay, so normally a single person will be handling all these techniques. ஏன்னா இப்போ நார்மலா மற்ற லேப்ல இருக்கிற மாதிரி 2-3 பேர்லாம் வந்து they will not be handling, only a single person will be handling all these things. Okay, so this is about micro manipulation technology. And the equipments used in micro manipulation technology. Okay. So next week, who's going to give a seminar? Lisith irukana? Lisith? Lisith illa. Arun Raja? Sanjay? Prashant? Prashant. Prashant. Next one, the Priyadarshini, Reshma. So you prepared for next week seminar.
Okay, sir.
Okay.
Sir, topic யார்கிட்ட சார் இருக்கு?
டாபிக் என்ன இருந்துச்சு நான் மாத்தி தரேன். 4th and 5th யூனிட்ல ஏதோ டாபிக் இருக்கு. நீங்க 3 பேருமே ரேஷ்மா, பிரியதர்ஷினி.
சார்
ரேஷ்மா இல்லையா? ரேஷ்மாட சொல்லிருங்க. ரேஷ்மா இருக்காங்களா? ரேஷ்மா?
எஸ் சார்.
நெக்ஸ்ட் வீக் டாபிக் நான் சொல்லட்டுமா இல்ல நீங்க எடுக்கிறீங்களா?
டாபிக் சொல்லுங்க சார்.
ஓகே, டாபிக் சொல்லட்டுமா? நான் உங்க சாய்ஸ்ல விட்டா நீங்க செலக்ட் பண்ண மாட்டீங்க. So artificial insemination and germ cell manipulation. யார் எடுக்கிறா? பிரியதர்ஷினி?
ஓகே சார்.
ஓகே. ரேஷ்மா?
சார்
you take micro manipulation technology.
ஓகே சார்.
And breeding of farm animals. Okay, அது எடுங்க. IVF and embryo transfer. பிரசாந்த் நீ எடுக்கிறியா?
ஓகே சார்.
ஸோ நீங்களும் எடுத்துட்டே வாங்க, நானும் அப்படி நெக்ஸ்ட் வீக்ல அப்படியே எடுப்பேன். ஓகே. அப்புறம் இன்னொரு 3 பேர் நேற்று சொன்னீங்கல்ல, ரவீனா?
ரவீனா ஆப்சன்ட் சார்.
ஆப்சென்ட்டா? இன்னொரு 2 பேர் யாருமா? கபிலாவா? கபிலா தான் வேற இருக்கு.
ராகவி சார்.
ஆமா, ராகவி. இல்ல கபிலா வேண்டாம். ராகவி இல்லையா? ராகவி இருக்காங்க. ஓகே, ராகவி நீங்க என்ன டாபிக் எடுக்கிறீங்க?
சார் நீங்களே சொல்லுங்க சார்.
ஓகேமா. Enrichment of X and Y bearing sperms from the semen samples. அது எடுக்கிறீங்களா?
ஓகே சார்.
ஓகே. அண்ட் இன்னொருத்தர் வந்து கபிலா.
சார்
கபிலா, நீங்க அந்த கேரியர் இன் பயோடெக்னாலஜி நீங்க சொல்றீங்களா? கேரியர்ஸ் இன் பயோடெக்னாலஜி
ஜெனரலா தான சார்?
வேண்டாம். கேரியர் இன் பயோடெக்னாலஜி இப்போ எப்படி சொல்லலாம்? ஏதாவது கம்பெனிஸ் பத்தி சொல்றீங்களா?
ஏதாவது கம்பெனிஸ் நீங்க அன்னைக்கு சொன்னீங்களே சார், அந்த
கம்பெனிஸ் மட்டும் இல்ல, எனக்கு ஒரு 2-3 கம்பெனிஸ், இந்த கம்பெனிஸ்ல எப்படி அப்ளை பண்றது, அங்க என்ன பண்ணிட்டு இருக்காங்க அப்படின்றது கொஞ்சம் சொன்னீங்கன்னா.
ஓகே சார். இங்க தானே சார்? சென்னை தானே சார்? சென்னையில
சொல்லுங்க. உங்களுக்கு எங்க உங்களுக்கு தெரியுமோ இல்ல உங்களுக்கு தெரிஞ்சா வேற ஏதாவது சொல்லுங்க. இல்லாட்டி சென்னையில் இருக்குது சொல்லுங்க.
ஓகே
கம்பெனி மட்டும் சொல்லுங்க. ஓகே
சார்.
ஷரத் இருக்கானா? ஷரத் இல்ல. ஆஜித் ஷரத் 2 பேருமே இல்லையா? ஆஜித்
சார்.
ஆஜித்
எஸ் சார்.
நீ என்ன எடுக்கிற? 51 தான் எடுக்கிறியா நீ?
ஆமா சார்.
கான்செப்ட்ஸ் இன் டிரான்ஸ்ஜெனிக் அனிமல் டெக்னாலஜி. ஓகே. Yashi and Akshaya.
Sir? Yes, sir.
Yes. So, Yashi and Akshaya, higher studies in biotechnology.
Okay, sir. Okay, sir.
Okay. Okay, so இது வரைக்கும் இருக்கட்டும். காமாட்சி இதெல்லாம் அப்புறம் பார்க்கலாம். Wait a minute, get the attendance. Ajit.
Yes, sir.
Yeah.
Yes, sir.
Anucha absent. Arun Raja absent. Everton.
Yes, sir.
Present, sir.
Kavila Kumari.
Present, sir.
Joel.
Present, sir.
Kamachi.
Present, sir.
Visit. Visit absent. Nitish. Okay, Lakshmi. Raghavi.
Present, sir.
Okay. Raghavi.
Present,
Reshma.
Sir.
Yes, yes, okay, okay. Sanjay. Absent. Sandhya.
Present, sir.
Sharat absent. Yashi.
Present, sir.
Aslima. Avina absent. Dikshita.
Present, sir.
Sadia.
Present, sir.
Shruti. Shruti.
Yes, sir.
Surya Pradapan.
Present, sir.
Antika.
Yes, sir.
Priyadharshini.
Present, sir.
Suganya.
Present, sir.
Okay.
Sir,
Yes.
sir, Bhavya present, sir.
Okay, present nama. Further information. Again, we are going to NAC. Actually, we have A grade now, and we are going for A++ grade. So again, we are appearing for NAC. So in the 15th we are uploading for NAC. So A++ we are getting ready. Okay. Okay, ma'am. Thank you.
Thank you.
Okay. Next week.